Ready? Okay, good afternoon, everybody. It is a very great pleasure to introduce Ron Evans, who we should have been having regularly as a seminar speaker, but are privileged to have today. Ron got his both his bachelor's and his PhD degree at UCLA in bacteriology and microbiology, respectively, and then went to do a postdoc with Jim Darnell at Rockefeller. And he did that, finished there in 1978, and was immediately given a job at the Salk Institute as an assistant research professor. And he shortly thereafter became a Howard Hughes investigator, very long time, 35 years as such, and was inducted into the National Academy of Science in 1989, and is currently at the Salk Institute, the March of Dimes Chair in Molecular and Developmental Biology, and a professor in the Gene Expression Laboratory. He has received multiple awards, many too many to name. Recently, one from the AACR. I don't know the name of that because that wasn't on his CV. Okay. Also recently, the Assan Award, the Nomis Foundation Award, a Charles Best Award in diabetes. Over the past years before that, he won the Dale Medal, the Wolf Prize, the Lasker Award, the Keogh Prize, and probably nearest and dearest to our hearts, since this is a pharmacology seminar, the most meaningful first award that he got was the Goodman and Gilman Aspet Award. So that kind of inducted him into pharmacology. He's also an adjunct member of the department. He's given tons of keynote addresses, named lectures, committees, and boards. He has more than 450 publications. I started to count the ones in Cell, but I was unable to. I had to stop. Cell Science, Nature, most of them, 450 of them, 100 or more reviews. He's also worked with probably maybe a quarter of the people who are on this Zoom or in this seminar, and some of the people who are students in our department. He worked with Jeff Rosenfeld and Susan Amara a great deal, with Wiley Vale, with Marion Waterman, with David Mangelsdorf, who notably was not one of our students, was a postdoc with him, but is the chair of pharmacology at UT Southwestern now, Chris Glass, Rusty Gage, Jerry Olevsky, David Brenner, Bob Tukey, Alan Saltiel, to name but a few. So if I didn't name you and he worked with you, you're probably listening, and he has been a really great collaborator. I think I'm embarrassed to not have worked with him over all those years. His interests and research has been wide-ranging, but focused on the steroid receptor family, which a field that he basically invented. He was involved in, has been involved in basically characterizing the nuclear receptors for the whole range of families, working on the retinoic acid receptor and quite a lot on the PPAR alpha, gamma, delta receptors, which got him into atherosclerosis, diabetes, exercise, and running. Scattered throughout his CV, he's done work on cancer as relates to the steroid receptors, liver cancer, intestinal cancer, pancreatic, and now a new area that he is going to tell us about today is colon cancer and its relationship, I believe, to some of the steroid receptors. So it's a great privilege to have you, Ron, and I'll let you take it from here.
I'll use this. Yeah. It sounds like it's working. Joan, thank you very much for the kind introduction. And it's really great to be back in person, to be able to talk to friends and colleagues and students directly. It's so much better to be in person when we can do it, but also welcome to everyone who's online. I'm going to talk today about you know what I absolutely need. Oh, there it is. Sorry, timer here, so I can make sure that I don't go more than a half hour too long. Today's talk is going to be about colon cancer, and the title here is "Colon Cancer Checks In When Bile Acids Check Out." So that's a little bit about the summary of what I'm going to be talking about, but I'm going to give you a little bit of background as well, especially in visiting the Department of Pharmacology, and I'm getting a little bit of my advancer is not advancing. I can click at this thing. Okay, so what I want to start is with this little cover example of the nuclear receptor superfamily using the Rosetta Stone that was discovered in the town of Rosetta. Some of you may have seen this in the British Museum, but we've kind of sort of altered it because remember the original Rosetta Stone was to link 3 different languages, and with the way in which each language writes it, and so it was really a major conceptual advance of how to translate between different communication systems. So now we just have it here at the top of this listing nuclear receptors like AR, TR, RAR, LXR. These are members of the nuclear receptor superfamily. And then below that are the DNA binding sites. You can see T, G, T, C, 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 T, G, and these are all the different binding sites for the nuclear hormone receptors. And then below that are the ligands. And you can see there's aldosterone, glucocorticoids, progesterone, thyroid hormone, and many other types of vitamin D, for example, vitamin A. And so this is a way of thinking about how you translate the language of physiology from what you can measure in blood and in physical ways to a genomic basis. And so, and when we look at physiology, we look at a set of interactive genomic programs that use ligands to communicate throughout the body and to coordinate these genomic networks. And so what you're really doing with hormones that are being delivered in real time is activating gene networks in specific ways in specific tissues for a specific duration. And depending on the amount of hormone that's being produced, that will also be dynamic. And so there's a lot that's going on in the way in which underpinning of this aspect of body physiology works. I have to use this other funny click here. Oh, sorry, I want to go back to Claude Bernard, who was one of the great early leaders in physiology. Claude Bernard wrote the book, The Principles of Physiology, which had a major impact. And he talked about the concept of physiology not being a one-way system, but rather being a dynamic process in the body. And that basically, if something goes up, if glucose goes up, then it will come down. And if sugar goes up, it will come down. And the idea of the maintenance of body functions, which he termed homeostasis, which became basically the underpinning of the dynamic nature of the way in which the body works. That ultimately led subsequently to discovery of the early hormones, which were back in the time around between 1910 and 1925, basically. This is Edward Kendall, and discovered 2 of the more important hormones, thyroid hormone or thyroxine, 1914, and he did the crystal structure. He announced it in 1915. He happened to get the structure slightly wrong, but he did discover it, and really opened up a new era of having a small molecule, which is basically a modified amino acid, having a hormonal property of controlling thermogenesis in metabolism in the body. He also isolated the first glucocorticoids and became very well known for that. Crystal structure of glucocorticoids was completed by Thaddeus Reichstein, shown here, won Nobel Prize for this work. And then in addition, after that, Adolf Windaus, who discovered that vitamin D, which was also isolated in this time, he didn't isolate vitamin D, but discovered that vitamin D was highly related to bile acids. And that led him to say that bile acids are sterols. There's an L missing in that, you can see, in 1919. So you can see what was happening in this window of time that was changing the dynamics of the way in which we think about the fundamental features of physiology being controlled by small molecules, particularly lipophilic molecules, some related to each other, such as sterols and vitamin D, some not. Like thyroid hormone is completely non-steroidal, and retinoic acid, vitamin A, as well in that time frame, also not steroidal, although it does come from the mevalonate pathway. So getting back to the theme of the talk, I want to talk about one of the particular receptors that we discovered in 1995 called the farnesyl X receptor, or FXR, and I'll talk to you about that in more detail, and its potential role in colon cancer. This is work that was done by Ting Fu in the lab. The underlying theme for this talk is trying to understand something that relates to cancer, but is elusive in most diseases, and that is risk. And the idea of risk is that you can measure risk in many diseases. For example, obesity is a risk factor for colon cancer. That's known. I'll show you a little bit about that. And there are many risk factors for, it can be heritable risk that you can have for certain diseases. Risk means that you have an increased potential to get it, but doesn't mean that you are going to get it. And you can measure that risk if you want to do it in terms of obesity per pound. And that risk changes specifically as you go up or goes down. But it doesn't say anything about the mechanism of what risk is or why risk is something to worry about or manipulate. And you can't really work with it without understanding what risk is. And so we began to want to explore the underpinnings of risk, which has many kind of interesting and complicated equations to come up with what a risk factor is. But what really is it, and why is it important? So I'm going to use the intestine just as a way to exemplify this. And I don't know if you can see my arrow is not very, huh? Oh, it shows, okay, good. So these are risk factors for colon cancer, but you'll recognize there are risk factors for many other diseases as well. So sedentary behavior and high-fat diet, and there's kind of one of the major drivers of that. And obesity is, which is a consequence, and you can correlate all these things together. Inflammation can be driven by this process, but in general, intestinal inflammation, however you achieve it, is a risk factor. Family history, age. Again, what does age, what's a risk factor mean in regard to each of these things? And so what does risk look like? And I'm just going to show you the way in which it's often measured. This is looking at just the United States, and what it basically shows is that obesity is a regional and somewhat geopolitical factor linked to high-fat diet. And it turns out that Colorado is the lightest state. Mississippi is the heaviest state in terms of weight and obesity. And if you look at this, basically same map, but it's a different map. This is the risk factor for colon cancer. And you can see the same trend. And this is the reason that you can say that, you know, where you live and where you eat and what you eat and your weight are linked to this cancer risk. And so this gives us a little bit of an underpinning of the importance of diet and exercise, but it doesn't really say what is going on. I want to talk about that. And to do that, just give you the simplest, what's called the Vogelgram sometimes. I'm not actually, after having done some of the work that we've been doing, as much a fan as I was of this before we started doing the work. The Vogelgram kind of simplifies making cancer a linear process. The normal epithelia, the adenomatous polyposis coli gene, or APC, is the mutation for almost all the animal studies in colon cancer, and it's about 80, at least 80 or 85% of patients that get it. Humans have a mutation in the APC gene that gives an adenoma. It does not give cancer. It just increases proliferation from the crypt. And where's my little pointer here? Hello, there you are. Okay, and then, but with second mutation with KRAS, you then get increased proliferation. This is a late adenoma. Still, it's not cancer, but now it's actually detectable and these can be removed. But then with the third mutation, P53, cancer. So now you have this progression between these genes, and then it's still localized and potentially treatable, but then there's additional progression with other changes to metastasis. And so this often happens in early years. One of my, Miriam Madian, one of the postdocs in my lab, and ultimately began working with Alan Saltiel here. She contracted colon cancer when in my lab and managed to have a pretty good run of finishing her postdoc, getting married, joining Alan's program. Eventually we lost her, but much of what we're doing has in mind that she was not obese or any other of the common risk factors. And so it's really important to know what is driving the nature of these disease progressions. And I'm going to just kind of sort of continue this. At this stage, and when we began this, it's Pandora's box. A lot of things are happening, and it's hard to tell once you open this box and things start coming out, what is the key feature? And one thing about Pandora's box is when all of the evil things leap out, like a P53 change or whatever it might be. One thing that the only one of the fates that doesn't leave the box is hope, which I thought was an interesting thing in looking a little bit in the history of Pandora's box. And so I want to just sort of expand on this concept just as a way to have a visual way to think about what risk might look like. And we use these cogs in a wheel to basically simplify the concept of three components: H, or hereditary risks, and that's like such as Lynch syndrome and colitis. These are if you have familial risks. Environment is what you eat. That's basically, and the things that you bring into your body that have to be contributing to this. That's like the Western diet. And then the third part is replication. Something has to increase the proliferation rate and change that. And that is the other part here. And that really is the Pandora. What is happening to understand how these components are coming together? So we're going to be talking about the H, E, and R of this, so the hereditary, the environment, and the replication components, see if we can begin to triangulate risk in the context of cancer. So this gets back to now, the reason that my lab got involved in this is in 1995, we discovered Barry Foreman and Carrie Weinberger began working on a receptor that ultimately we called FXR. It was an orphan receptor at that time that Carrie had isolated. And we began trying to understand what it is. And ultimately, and I'll just tell you that it came out to be a bile acid receptor. And that was a surprise. At that time, bile acids had one thing. Alan Hoffman basically was a leader of the field. And it was the cataloging of bile acids in all, if they're present in all vertebrates, all species. You can measure them in lizards and worms and frogs and everything where you've got. I'll take out the worms. Basically every animal could be cataloged. And there was such a dynamic field in trying to do this and trying to understand what they were, which basically people put them as surfactants with special properties to bring in lipids and cholesterol into the body in specific ways. And they're a transport system, and they actually are dynamic because the more lipid you eat, the more bile acids you need to make. And so there's a kind of a conjoint link between high fat diets and bile acid pools. And so this had a big impact, but then they did so much analysis in the field kind of cooled down and became a minor field from being a major field. Remember, it started back with bile acids winning a Nobel Prize for just being related to vitamin D. Now we learned everything about it. And the discovery of FXR as a bile acid receptor and a member of the nuclear receptor superfamily said bile acids are sterols, which we knew that they were, related to vitamin D, which we knew what they were, but more generally, these are hormones. And not only are they hormones, they vastly outnumber all the other steroidal hormones in members of the nuclear receptor superfamily by far. And so if this is the correct interpretation, then it's a bile acid receptor that creates a different view of the role of bile acids in body physiology, and means that the bile acids are informational, they function as hormones, and they control gene networks. And if you look at this map, this is the adrenal gland, this is the kidney, super high expressors of the bile acid receptor. Also the liver, very high, and that's where bile acids are produced. They're stored in the gallbladder, ready for delivery with food, highly expressed in the intestine and pancreas. So all these are very high expressors. These are not the only tissues that express the bile acid receptor, but they are dominant ones. The one thing I want to point out before leaving this slide is chenodeoxycholic acid, classic member of the bile acid collection. And there are quite a few about 35 or so typical bile acids in humans, as is true for mice and other animals. So it's an elaborate collection. This is a natural ligand, a natural bile acid. This is the drug we made at the Salk in my lab. Michael Downs generated this called FXR, that links to the receptor, FXR, amine. It's a completely synthetic compound that activates the bile acid receptor as if it was a ligand, as if it was a bile acid, but it's synthetic. And it doesn't have any of the properties, stereochemical properties of bile acids. It's completely a chemical that is on its own. And so it's not a bile acid. This is intestinal specific. If you give it orally, it gets into the stomach, gut, and then it's pooped out. It does not get into the bloodstream. And so when we use this drug, which you'll see several times, it's only activating FXR in the gut. So this just raises the question quickly, is how do bile acids and colon, what the link between bile acids and colon cancer? And some of the questions that we're going to address is, how does FXR impact intestinal homeostasis? That's key. And how do bile acids modulate intestinal physiology? How does high-fat diet alter the bile acid pool? How does high-fat diet accelerate colon cancer? And can we use what we learn to develop new ideas and concepts in therapeutic approaches? So if we just look simply at the diet issue, I'm going to basically tell you that if you feed mice on a chow diet, you're looking at the kind of the histopath colon, and you can look at just the villi and how the length of the villi. And if you switch from a chow diet, normal diet, where you have healthy intestinal state, and go to a high-fat diet, you see the villi, it's a different direction of the arrow, but it's much shorter. That's what happens with inflammation. Is being driven by the high-fat diet and the increased dynamic pressure of trying to take in all of the lipid, which much of it stays in the intestine longer because you have to take in more. The bacteria want that. And bacteria are very good at modifying bile acids to actually become, instead of good ligands, I'll show you this in a moment, for the bile acid receptor, to poor ligands or antagonists. And the point of this is high-fat diets trigger a process that alter intestinal physiology and function. So look about what is the nature, the link between structure and function in this process. We can ask is, how does disruption of this structure affect physiology? And this is really kind of two pieces of data here. One is just looking at serum bile acids on a wild type diet. Let's say that's you or the mouse. This is a normal diet, relatively low levels of serum bile acids. Remember, this is what's in your bloodstream after you've released it, you absorbed it back. If you're on a high fat diet, you can see you're more than double your bile acid pool, more than double what's normally in your bloodstream. Interestingly, entirely unexpectedly, and I was really excited by this particular result here. A normal diet, but with APC-min, you have increased the bile acid pool by 4-fold. Increasing anything, think if it's your salary, but think if it's your weight or whatever. In physiology, 4-fold increase is a big change. And this is in no high-fat diet. So this means a genetic mutation in the single most important driver for colorectal cancer in people. The mutation in that gene leads to a direct change in the production in the liver of bile acids and the bile acid pool, and resulting in a major bile acid change. So we have a genetic change that is now linking to what? Bile acids. So this is why bile acids now become more interesting, is it's directly linked to the major driver of this disease. Now, when you take an animal that now it has the APC-min mutation, you now look at the change in serum bile acids, and it is now 8 or more fold increased. So now you have this conjoint relationship between the mutation in bile acids and the diet, high-fat diet in bile acids. So you have two convergent components. And so these are physiologic pathways. And so I call this with the, and I'll show you a little bit more, and then I'll sort of go into this in more detail. A different way to measure what's happening in the gut is looking at something called leaky gut. We do this by looking at fluorescent markers. And again, very little in leaky gut in a normal diet. High-fat diet causes leaky gut, is here. You can see maybe a 3-fold change. The mutation causes leaky gut, again, and high-fat diet compounds that process. And so we have a series of events, and we name this physiologic convergence when the diet and the genes are operating cooperatively. And then pathologic convergence when you have a mutation and a high-fat diet, an abnormal diet. So you have two different states, physiologic convergence and pathologic convergence. These are terms we made to kind of collectively give a concept now is what is underpinning what we might call risk. And so you can now begin to bring this down to specific things that you can measure. And also the surprise that was unexpected is this link between a mutation and something like a physiologic mechanism such as bile acid production being hijacked in a very profound way. So we can then begin to get a little bit more granular by looking at the way in which bile acids our individual bile acids are changed. And I'll just tell you specifically that one known pro-inflammatory bile acid that has been known for decades actually is taurobeta-muricolic acid, TBMCA. And you can see that not only is it known to be pro-inflammatory, but it also changes just in the context of diet. And so on a normal diet, it's at much lower levels. You can see the levels, and then it's a high-fat diet, it goes up again rather dramatically. And if we link that with the APC min mutation, look what happens to torabeta-murocholic acid. Again, this mutation is specifically in, hard to even imagine the change in this specific known inflammatory bile acid. The mutation is causing this such a highly increased alteration that it's going to be hard to escape the impact of these alterations in the bile acid pool and maintain normal homeostasis in the gut. And so all this fit together, and kind of you can visualize what is happening and conceptualize what it is now, I think, and measure it in the components that are actually underpinning what may be some of the pathology. But keep in mind, these are all acting concurrently through the bile acid receptor, FXR. And I'll show you some of that. And so when we look at what is happening and how these species and the events that are concurrently happening are manifesting themselves, we can become a little bit more granular in how we measure things. This is very quick. If you look at the number of tumors in the APC min mouse and correlate it to the change in torabeta-murocholic acid, you can see that it directly correlates with what is happening in these animals. It increases with the number of tumors. And so the question then says, is torabeta-murocholic acid the mimic? Is it the actual physiologic readout for what is happening in the inflammatory components, is what's happening in the gut? If you just give torabeta-murocholic acid to a normal mouse on a regular diet, what is that going to do? And so we did a number of different studies to look at torabeta-murocholic acid. And basically, as I'm quickly through some of this, is that it is sufficient to drive the system cancer and it enhances many stem cell markers. These are all cancer stem cell markers that get elevated just by giving torabeta-murocholic acid. It also suppresses FXR levels itself, suppresses another bile acid response regulator, which I didn't talk about, called CHIP, and IBAP, one of the bile acid binding proteins, the illegal bile acid binding protein, also suppressed. And so you're suppressing bile acid physiologic management, and you're getting now increased changes in stem cell markers and LGR5, which is one of the markers for the crypt, factomedin, many different Wnt3a. These are all markers that are being elevated. And so this is just by giving this one bile acid. So now we can link the ability of the bile acid to trigger specific inversion of normal physiologic patterns. So it's in this pathologic convergence state. And essentially it inhibits FXR action in vivo and activates these more proliferative pathways. And I'm tell you a little bit more about how that's happening here. This is just looking at a mouse that's getting a hit of torabeta-murocholic acid, and oh shoot, it didn't. There's a do this. I don't know, maybe it's, well, okay, it's not giving me, there's a build here that shows that this gets nastier and nastier. I'm not sure why it didn't come up, but you can see the consequence of taking torabeta-murocholic acid on EDU incorporation, that is DNA damage and then incorporation of EDU, which we can read out. And so basically, the more you increase torabeta-murocholic acid, the more EDU incorporation there is and the more proliferation there is. So now we have a direct link between bile acid and now proliferation. And so that further drives the concept of risk. And I hope that little squiggly things down here don't mean that the next slides are going to be squiggled up. Let's hope. Don't know why we're getting this lower panel down here, but okay. So if we want, one of the things that is interesting to see, besides what we use the term inflammation, but you can get more granular with inflammation and look at components such as IL-17, which is a known major driver of intestinal inflammation, but also systemic inflammation. So you can go from the colon to disease progression to the colon and to the whole body. And basically what this shows is the changes of this particular cytokine in the context of APC-min mice on normal diets or high-fat diets. And you can see we have to change the scales on many of these to be able to show the data. But the important part is now we're going to bring in the ligand. I remember I talked to you about FXR, which is a synthetic activator. So it should be like the inverse of torbeta-uricolic acid. And what we see is, if you look at the yellow and green, is whenever you give torbeta-uricolic acid, you bring down IL-17, as you can see there in these two components as well, in the APC-min mouse on high-fat diet, and then a different model as well for looking at IL-17 regulation in these various models. And I'm gonna just give a little interim summary now of looking at the different components of the cascade, is that we see that there's a series of events that are related to mutations, hereditary and environmental. They're interacting most likely with the FXR to either control or destabilize intestinal physiology. This is the genetic part, APC-min. This is the diet part, E. We're going to then trail down to what we're producing as the readouts, physiologic readouts that go here, torbeta-uricolic acid and also deoxycholic acid, which would be in people. The M here means it's a mouse. Leading down to a cascade that activates FXR, but the nature of the pathologic convergence is it takes FXR out of play. And in that context, without FXR driving this pathway, you really start to get out of control in body intestinal physiology. So we have the dietary risk triggers this cascade of specific, measurable, and quantifiable events that also have underpinnings of mechanism to begin to dig into this in much more detail. And I don't want to spend a lot of time in this, like there's a lot of different data, but we do want to make an example of how this synthetic ligand in the context of both the APC-min mutation and high-fat diet, that's the adenocarcinoma component. And I'll just say FGF15 is a direct target that gets activated during a meal. FXR activates directly this gene. It's FGF19 in humans. There's a clinical trial now going on with FGF19. It's one of the first of the specific target genes that was identified by Dave Mangelsdorf and Steve Cleaver and our lab together in a joint paper, that FXR is a direct activator of this. And now this is something that's in clinical trials and likely to get approved. And if you just look here for an example that of what FXD can do, FXD is going to be here in green, there's APC min, and then here, and so you can see that basically APC min drops the FGF15 to a low level, and when you give FXR, you can actually dramatically increase production of this physiologic and not in low inflammatory a molecule that you want to be present, but that gets suppressed dramatically in the APC min adenocarcinoma and even in the chow dye. You can see that. You can see the rescue. So if we just keep moving, we're manipulating these. If we just go to the bigger picture of the integration of all these problems is with colon cancer. And so we want to look and see what might be happening in more specific ways in that context and the nature. Remember I told you about leaky gut. Okay, a little music here. Okay, so we have in the wild type, the relatively low leaky gut, and the APC min on a chow diet, you see, a dramatic increase. But when you give the FXR ligand, you can see this dramatic decrease. And so it is working at least on the leaky gut component and also on the high-fat diet side as well. So this is one molecule just acting in the intestine that is rescuing the phenotypes that we're getting. If we look at the bile acid pool, these are the two models. I'll just go over here. You can see what happens in the APC in mouse and serum bile acids, which go really high. And you can see what happens when you give the drug. This is stellar. So now we have clearly a way by putting these different components together to collectively bring each of those components to both a hypothesis that by using therapeutic targets such as FXR and the addition of a synthetic ligand that's just present in the gut, you can reverse many of these symptoms based on transcriptional regulation of this intestinal network that's having a vast impact, not only in the intestine, but in the entire body, because all these bile acids normally get in the body and act throughout the body. And so we can now visualize conceptually and mechanistically what might be happening. And I won't go through this in too much detail. Basically, on the highly reactive adenocarcinoma diet, you can again see, just looking at the impact on torbeta-muricolic acid, giving what FEX does, just this one single bile acid, which we triangulate on that as a monitor. And then the question is about survival. And this is a little bit harder to do, and I'm going to show you the data that we have. Let me see this. This is looking the same thing, looking at a different bile acid, deoxycholic acid, which we share with the rodents. And so, I guess I tricked myself here. Before getting to the survival studies, this is just now a update on the schematic here about the nature of the ability to regulate cancer progression and how does all these pathways happen. FXR is basically suppressing the impact of diet and suppressing the impact of the genetics to suppress the production of some of these potentially toxic bile acids that impact on the replication rate. And so got a slide out of order here, but I will tell you what this slide means. This is just looking at DNA damage and the ability for the addition of this bile acid to increase damage. And you can kind of measure that in a couple of different assays. And now we're getting back to what I was trying to get to before. And that is looking at survival curves. And here's how we do this survival study. FXR is given to mice or not on this schedule. They're on a normal diet and a high-fat diet, and then they get PhD or vehicle, and then we just look at survival. And basically what you see, and I should say colon cancer is the second most lethal cancer that we have. Lung cancer, and then pancreatic cancer is the third. And we also study pancreatic cancer. Point of this is that with the one single drug that is intestinally restricted, we do get an extension going from 30 weeks to 50 weeks on the normal diet. But I have to tell you that in this model, the mice are not dying from cancer. They die from the adenoma. The upper GI blockage is what really causes the loss of these animals. It rarely gets to the colon. I didn't actually mention that on the high-fat diet, unexpectedly not previously reported, is that high-fat diet gives you colon cancer as well. So you get upper GI and colon on the high-fat diet. So it's a more aggressive disease. So looking at this, what we have is on the high-fat diet, a less of survival, but this is also it's a different disease, and it's a much more inflammatory driven disease, but it still gives us two things. It does lead to an extension, number one, and it's a single drug that we're using. And number two, it gives us an ability to look at the gene expression changes between these two states. And we take the signature that we get from these two, then we go to a human database and begin to benchmark the FXR signature that we're seeing in these mice that are getting colon cancer to what we see in humans. And basically what we find is if humans have the signature that we're coming out with, that differential signature, they have a much lower risk and improved survival, as you can see. So that gives us some sense of actual hope that we have something very special that could be translatable potentially to people. And there are trials there are studies that are taking off to begin to look at this. So we're very excited about that. And the potential for bile acids to be used and to take advantage of the tools that we have and we display here to actually turn this into a mechanism. So I just want to end on, and somehow the clock went up. We'll still a little bit of time here. To have one coda, which is a little bit different. It goes back to when we talked about the discovery of bile acids with Windhouse. And so I call this bile acids back to the future because there's more here than we think. And we need to think about it more deeply. So I call looking back to map the way forward. And the idea for this is, while there was a huge effort to understand and classify bile acids, that was in an era when that's all you could do. And so what I'm suggesting is a new goal here that relates to bile acids, which is to create a comprehensive molecular characterization of what I call the mammalian biome. And something comes up here. And I call this biocarta, building the biome. And the idea is that bile acids are dynamic. They're really responsive to diseases, but also to your genetics and what you eat. And so they have a definable chemical composition. They function they have functional properties that can vary across time, diet, genetics, and disease. So the bile acid pools, as I've shown to you, are very responsive to food and the microbiome. I didn't go through much of that, but there's a lot we've done on the microbiome as well. And these bile acid species, many of them, but not all of the bile acids that we have, can bind to the bile acid receptor. That really kicked off this sort of new evolution and sort of a paradigm shift in thinking about what are hormones and how are hormones linked to diet, and how could hormones be directly linked to diet as surfactants, and also having the built-in ability to activate genomic networks throughout the body. It's a real example of evolution. So bile acid receptor, vitamin D receptor, bile acid receptor, pregnane X receptor, bile acid receptor, constitutive androgen receptor, CAR, bile acid receptor, TGR5. This is a cell surface membrane transmembrane protein, but it's a bile acid receptor. In ROR, the retinoid orphan receptor that we discovered back in 1989, David Mangelsdorf discovered bile acid receptor. And that also was shown to be a very good bile acid receptor controlling IL-17. So we have just a number of different ways to rethink about what the biome is and the nature in which bile acids have pervasive and local systemic and system-wide effects. So what I like to see happen, I call bile acid now a liquid organ. It's really operating in a different conceptual mode. It takes advantage of all the aspects of the classic endocrine tissue like the thyroid gland or the adrenal gland, but in a very different way. And it's used to actually saturate the body based on what you're eating and the time which you do that and the way in which you do that in a quite surprising fashion. So we have to just rethink about when we talk about physiological mechanisms and how the system is set up, ways to do it. And a simple way to begin is just, this is like, maybe it's like third grade now, but to break things down into the bile acids collectively, such as ascorbated uricolic acid or ketodoxycholic acid and different components against potential targets such as FXR. And does it increase FXR? Does it activate FXR? Does it suppress FXR? Does it have no impact on FXR? What does it do to the microbiome? Does it have impact? And you can give very simple 1 or 0 kind of components. Does it change lipids? Does it activate the vitamin D receptor? And so, and TGR5, you can begin to ask when you activate these components, how does it change these components? And so, and you can kind of expand it even more to look at TGR5, PXR, CAR, high-fat diet, APC-min mutations. You can really keep expanding both the potential generators and manipulators against the growing biome. In our lab, in the Rob Knight's lab, have been working steadily on finding new species of bile acids, new types of microbes that are specifically modifying bile acids by a half a dozen amino acid modifications that were previously not recognized and missed originally. And the reason I think a lot of was missed in this field is because they always checked bile acids in normal animals. No one really looked in disease context. No one looked at high-fat diet. No one looked at cancer. And if you had, you would have come into a different gold mine that we kind of have walked into and came out with more than what we thought. So this is a summary. Bile acids are early sensors of dietary fat. And intestinal bile acids can modulate the epithelium, LGR5, crypt, stem cells. Their genetic and dietary risk factors for cancer target this bile acid axis. This is what I remember: pathologic convergence. Pathologic bile acids can promote DNA damage, and FXR agonists can reverse that, repress things like Wnt, and curtail tumor progression. So this is the last part, last one. Risk. Deconstructing risk reveals really an unrecognized mechanism of convergent pathways that deplete or modulate FXR signaling. Suggests that FXR activation by synthetic ligands can duly block the oncogenic and high-fat diet cascades to restore intestinal homeostasis pharmacologically, and to enable new approaches to cancer therapy, conceptual approaches and real approaches. So these are the people who took up this cause here. Ting Fu now has her own. This was looking for a job, and she already has a job now. And some of the other members of the lab that have worked diligently over the years. This particular story began in 1995. We have a lot of local collaborators here that really make it a pleasure to be in this environment, and some distal ones as well. And so with that, that's the journey that we've had, and I'll be happy to take any questions. You get the mic. Mike gets the mic.
We've shown quite a while ago that with the inducible mean APC ablation model of Eric Fearon, that as soon as you disrupt APC, the barrier is disrupted, endotoxin and bacterial nucleic acid translocate activates TLR4, TLR9, and so on. You knock out the TLRs, you block all of that. The TLR activation induces IL-17, IL-23, so you knock out IL-17 or IL-17 receptor and you block the whole process. So, so the high-fat diet may augment the barrier disruption, but the effect of the APC mutation is so strong that it's hard to know what comes first.
Oh, so, well, yes, that could be true.
So I told Vogelstein that I agree with him that APC mutation come first, and probably the inflammation may come second, because the mutation would cause the localized inflammation by disrupting the barrier.
Yeah, oh, sure. I mean, yeah, go.
So that bile acid comes also from the lumen, right? So it's another thing that will translocate together with the microbial products. Yeah,
yeah. So any way that you can get to disrupting the barrier, and there are multiple things that can do it, but from the link between risk, and speaking specifically, that's why this is based on risk, is that you can take the word risk and the number and translate it into a quantifiable mechanism. And so it doesn't eliminate anything that you're doing, which is, and you know, there's, and much of what you're seeing in the mouse is upper GI, unless you put them on a high-fat diet.
In our model, it was
well, you're in your, of course, in yours,
yeah,
but in yours, because it's knocked out all the way through, right? So, which is a little bit different than the actual typical model. So, but there are different ways of getting at the same thing, including the induction of IL-17, which seems to be a major regulator, and the inhibition of FGF15.
Ron, so thank you. So excellent review of the history of bile acids. So in that regard, so you alluded to the fact that some of these bile acids will be acting on TGR5, which is a G protein couple receptor. So first question, a pharmacological aspect, does fexaramine also affect TGR5, number one? In the second, any of the effects that you see on bile acids produced in normal or in cancer models, they can be assigned also effects on the GPCR. And I understand GPCR is expressed in macrophages very highly.
I couldn't get all of it because of the
TCGR5.
TGR5.
There will be the G protein coupled receptors.
FXR doesn't directly activate or alter TGR5 levels, if that's what you're asking.
Yeah, I'm asking the bile acids will be also working on TGR5.
Oh, yeah.
So some of the effects that you are showing with the bile acids, which one of them are dependent FXR versus TGR5?
Okay, so I mean so there's, as I said, there's at least 5 different bile acid receptors in the gut. All right, and so FXR, we call that as the master, but really there are these 5 receptors. One's the TGR5. There's not been enough individual triangulation on each of these to make the comparison you want. We're trying to do that, but when you're looking at 5 concurrent things, you have to like come out and do components to it. So this is a little bit of a journey that we have to go through to really be able to answer why do we even have 5? So they're not going to all be doing the same thing, and it's also a little bit more difficult to study things like TGR5 in the mouse models relative to human, which is because they're not identical in the way that they get the cancer. So I think that you have to work with what you can do. But I look at the production of IL-17 as one of the key hallmarks that you want to look at as a driver of what's going on. But also other aspects, such as what Michael said, leaky gut, these kinds of things that are really critical and have long been known to be major risk factors for the disease, but without having any underlying sort of biology or molecular biology to understand it. So those they have to come together. I'm very interested in the concurrent change in regulation, things like TGR5 and intestinal FXR and the pathways when they conjointly regulate the same thing like IL-17. So there's a lot to do, but it's actually pretty specific experiments that you can do to address your question.
So, and specifically, the drug you were using, fixaramine, FEX, does it work on TGR5?
No. What was that? Oh, sorry. Oh, sorry. I was walking away from the microphone.
I guess I have to ask a stem cell question here. So you showed this data with increasing stem cell signals. So do you know if the receptors are heterogeneously expressed across the different cells in the intestine, or are the stem cells somehow more responsive to the signals or the activation of the pathways?
Are you talking about FXR or are you talking about all of the receptors?
Stem cell expansion or stem cell signal activation?
Yes, yeah, there's quite a bit of data that shows that if you increase the inflammatory, if you put them on a high-fat diet or you increase the inflammatory state, that you get increased stem cell proliferation and also increased damage of the crypt, DNA damage. The evolutionary of the crypt is designed to tightly exclude bile acids, and Alan Hoffman studied that. And so he was skeptical of this because bile acids can't get there. But in fact, they do, particularly in the inflammatory state. And they do damage. People who reviewed our paper at Cell wanted us to explain exactly how it does the DNA damage. I said, not going to do that. because that's a whole different kind of mechanism. But damage happens. We don't know exactly how it happens. And there is a history of bile acids can conjugate with DNA. But I don't want to, it's a different layer of what we're interested in.
Ron, a couple of questions from the chat. One by AC, whoever that is. What are the phenotypes of FXR mutations or deletions?
So the reduction or deletion of FXR relates to a very highly inflammatory intestine, number one. It's makes also extremely more sensitivity, changes dramatically your microbiome as well. I should say inversely, activating FXR changes the microbiome dramatically. And you can activate it, as you saw, on a high-fat diet. And that gives you a new microbiome. Doesn't give you the original microbiome, nor does it give you the high-fat microbiome. It gives you a pharmacologic, Joan's going to like this, a pharmacologic microbiome that's very specific with the drug. And so it's not like random changes, very specific changes to that. And so when we're restoring these physiologic mechanisms, we're not going back to the actual true baseline. We're going back to something that's pharmacologically induced, and it's entirely reproducible, and which is interesting to understand, but it's a way that you can show how the environment leads to adaptive changes that are not random, but really lead to, in some ways, what you eat. It's going to establish something unique but reproducible in individuals.
There was another question, but it related to a specific slide. So I don't think you're going to ask it. It was about what are the white drops in healthy epithelial cells, but we probably don't know what slide that related to. I have this is a sort of conceptual question. I guess I would have thought from the way you brought it all up that the receptor and the bile acids were stimulating the receptor and were pro-inflammatory. And I would have thought that the agonist was going to be a bad thing, the FXD. I mean, the story came out later, but basically those things are through suppressing, through the FGF and through IL-17 are having kind of suppressive effects.
That's correct.
So, yeah, okay. So,
Yeah.
Because, so the high bile acids are pro-inflammatory, are like, how are they different from FXD?
So
Yeah.
if you stimulate the increase in the bile acid pool, the problem is I didn't show you this, but if you look at the generation of the bile acid pool, it's a very complicated cascade. There's about, because you're producing 35 or more different related sterols, and that's asking the liver to do a lot. You have the adrenal gland. There's two steroids or gonads or one steroid they have to make, and they can change their levels dramatically. But when you're making that many, if you put a high pressure on increasing that by having a high fat diet, you can't process it properly. And so you get a lot of variant bile acids. These are the ones that were missed by people like Alan. And so these variants, because of the nature of the pressure on the enzymatic system, gives you all these diverse ones, but they're not, they don't have the evolutionary design to now interact with FXR in the appropriate way. So they're antagonistic generally, which is the default. And so that's why these high fat diets causing a problem, because you're generating pathologic whole collection at very high levels of bile acids. And so these fourfold pool changes or more really become a problem. But at least we know that, and we know you can now address it. And hopefully we'll have intestinal restricted human ones, drugs that just go through. And so very high safety value, but potentially very high payoff.
Okay, well, thank you so much for bringing us into this
Okay.
exciting new story. And we look forward to hearing more about it.
Okay.
And thank you for a wonderful talk.
Yeah. Thank you.